Okay, so in today's session, we're going to be talking about the structure of skeletal muscle. Skeletal muscle, sometimes referred to as striated muscle, so the muscle that's attached to your bones. So we know that there are three types of muscle in your body. There's cardiac muscle, which is the muscle found in your heart. There's smooth muscle. This is muscle that is controlled by your autonomic nervous system and is usually muscle that makes up things like your internal organs, like your intestines and your stomach lining, that sort of thing. And then you've got your skeletal muscle. So you can always identify skeletal muscle because it looks quite different under the microscope to the other two types of muscle. Let's just briefly look at how these structures are different for a second. So when we talk about muscles, muscles in our body tend to come in pairs. There are exceptions. So for instance, one exception is your tongue. Your tongue doesn't have an antagonistic pair. It's just one massive, really powerful muscle. And the other thing that's really interesting about your tongue, not just that it doesn't come in a pair, is that it's not attached at both ends. Most of your muscles are attached at both ends to your body, whereas your tongue is free at one end, meaning that it has much greater range of movement, and it also is really strong to enable it to do all the movement that it needs to do. So when we talk about muscles, we tend to say that they come in antagonistic pairs. This means that there are two muscles and they work against each other. So when one contracts, the other is relaxing and vice versa. An easy example for you to see and look at on yourself is your bicep and your tricep. So when you bring your forearm up towards your shoulder, your bicep contracts and you can see that, and your tricep relaxes. And then when you bring your arm back down, you can see that your tricep contracts and your bicep relaxes. So I'm just going to introduce you to a few key terms that are not really super necessary for your A-level understanding of muscle, but are just generally useful. So you may have come across the term ligament. Ligaments connect bones to bones, and tendons connect muscles to bones. So tendons connect our muscles to our bones, and wherever two bones meet, we call that a joint. And at the joint, we find cartilage. So cartilage is a soft, spongy connective tissue which cushions the joint and prevents friction and wear and tear between the bones. So you might come across the words ligament, tendons, joints and cartilage, and these are all just general terms that you need to understand what's going on with our muscles. So the job of our skeletal muscle is to move our bones. So muscle tissue is highly specialized tissue and it's made up of muscle cells. But muscle cells don't really look like we imagine cells to look. They don't really look like your average cell. This is because they are highly specialized to allow contraction. So they're essentially just packed with these strips of contractile proteins to enable the whole thing to be able to contract. So we have this quite complex structure. Let's have a look at it here. So here you can see the muscle attached to the bone via tendons, and then we can see that the muscle is formed into bundles of muscle fibers. And the whole bundle of muscle fibers is surrounded by connective tissue. So if we were to cut this open and have a look inside one of these singular bundles, we would see that this cell now inside is very strange looking to us. All of the organelles are pushed to the very edges. So we can see the nucleus pushed to the very edge. And then we can see what is actually a massively modified endoplasmic reticulum. But this is an organelle that you've never come across before. And in muscles, this is called a sarcoplasmic reticulum. So this is a highly modified endoplasmic reticulum. It performs a really specific function within a mature muscle cell that we'll learn about later. And it surrounds the outside. Okay, so we're going to look at it in more detail in a minute, and you'll see that it sits around the edges of each of these bunches of what we call sarcomeres, which are the individual units of contraction. So you're probably noticing this word sarco pop up a lot. So sarco is one of the prefixes that we have with muscle cells. You can see sarcolemma is the modified plasma membrane. So sarcolemma is really just the name we give to the cell membrane of a muscle cell. And then sarcoplasmic reticulum is just the name we give to the endoplasmic reticulum in the muscle cell because it performs a really specific function. And then can you see between the sarcoplasmic reticulums at the edges, we've got these tubes called transverse tubules. These are going to be really important. You'll learn about these later when you learn about how nerve cells or how motor neurons interact with muscle cells and get them to contract. So the transverse tubules are involved in that process. So then if we look at one, can you see now this one individual fibre of muscle tissue here is called a myofibril. Again, myo is a common prefix in muscle cells, so you've come across that before when you learn about myoglobin. So one single myofibril is made up of many sarcomeres all sat next to each other. And a sarcomere is this, it runs from one Z line to the next. So we are going to be focusing now on one sarcomere. Just take a look here at this image for a second because I think this shows it even more clearly. So can you see that the myofibril, the longitudinal sections here, and you've got one sarcomere and then the next and then the next, and then surrounding each sarcomere you've got the organelles of that cell. So you can see you've got the mitochondria, you've got the sarcoplasmic reticulum that we mentioned before, and you've got the sarcolemma, which is the modified plasma membrane, and then this really clearly shows you how the T-tubule is an extension of the sarcolemma. So the T-tubule is how the sarcolemma is able to run throughout the whole of this myofibril. That will be important when you're learning about how motor neurons interact with muscle cells, but essentially it's because you want each section to contract. So each section, each sarcomere needs to receive that information to contract, and so this membrane is just a massive extension of the membrane. So let's focus on a single sarcomere. So look, the sarcomere runs from the Z line to the Z line. Let's look at this single sarcomere. So if we look at what's actually going on here, one single sarcomere, so one unit of contraction, is made up of a number of different proteins held together. The two main proteins that we see here are actin and myosin, and we'll look at the structures of actin and myosin separately, but let's just look at how they sit together. So the ends of each sarcomere are known as the Z line or the Z disc, and these are made up of a protein called titin, but actually you don't need to know about titin for your A-level, so don't worry about that one too much. You've then got actin filaments coming off the Z line towards the center. The center of the sarcomere is called the M line. And then in the center, pointing towards the Z disc, you've got the myosin filaments. So the thick filament here is made up of many thousands of myosin molecules all joined together, with their tails attached to the M line and then their heads pointing outwards in all directions and binding onto the actin molecules. It's really important that we can imagine this three-dimensionally, because this is a three-dimensional thing, And it's also really important that we can understand this as a process. So all these things are going to be creating movement, going to be creating contraction, and we need to understand how they do that. So we can see the thin lines in the diagram are made up of actin, and the thick lines are made up of myosin. The tails of the myosin meet at the M line. And then what other names do we have here? The H zone is the area that contains only myosin filaments. The A band contains both actin and myosin, and the I band contains only actin. Take note that the I band crosses the Z line. So we've got the H zone, the A band, the I band, the M line, and the Z line. So these are some names that you need to know, and you need to know what they are made up of. Let's talk about myosin in more detail. So myosin makes up the thick filament that we see when we look at a sarcomere down the microscope. It is a fibrous protein, and it is made up of two molecules wound around each other. Can you see how this myosin molecule is two of them, two tails and then two heads poking out in either direction? So this fibrous protein has a long tail, and then it has a head, and that head is able to attach to the actin filament. It's able to do that because it has an actin binding site, so a site on it which is complementary to the binding site on the actin. It also has a myosin ATPase site. So this is where it can interact with ATPase to break down ATP to provide the energy for this process. So we know that muscle contraction is an energy intensive process, so it requires ATP, and we'll talk about all the points within muscle contraction that require ATP later. Myosin is arranged so that half of the heads point in one direction and half of the heads point in the other, and the tails meet at the M line. Let's look now at actin. Actin makes up the thin chain of the sarcomere. So if we actually look at actin, it's a globular protein. So an actin filament is made up of many globular proteins which are arranged into this thin chain. And can you see how it's two of these thin chains of the actin globular protein wound around each other? And I think it's actually pretty amazing that in this electron micrograph you can see the shape of this actin filament. So the two chains twist around each other, and the ends are held in place at the Z line. Now, when we actually look at this thin chain in the sarcomere, there's actually more than one protein present. There are three proteins present. There's the globular actin, and then there's the fibrous tropomyosin. Can you see how tropomyosin is this very thin filamentous protein which just travels along the actin? Can you see how tropomyosin fits between the actin chains and it wraps itself around? The tropomyosin, when the muscle is at rest, is covering the myosin binding site on the actin. So that little black circle you see, that's the myosin binding site on the actin, and it's being covered up by tropomyosin. At regular intervals along the actin chain, we see another globular protein. This globular protein is called troponin. It's called a troponin complex because there's actually three globular proteins attached to each other, but it's fine for us to just know it's troponin. So in our thin chain, we've got three proteins: actin, which is the main one, tropomyosin, which is filamentous, and then troponin. So let's try and bring all these things together. So just have a look here at this sarcomere under the microscope and notice how we would label each part. So we've got the Z lines here. Between the Z line is the sarcomere, and we know the Z line is made of titin. And we know that the titin holds the actin in place, and we know the actin forms the thin chain and that it's made up of globular proteins joined together. Then in the very center, we've got the M line. We know this is where all the tails of the myosin join. Then we have got the H zone. We know that the H zone contains only myosin. and then the A band that contains both actin and myosin, and then the I band that contains only actin and crosses the Z line. So it's really important that we can label each of these parts and also that we know what's present in each of them. So what is the sarcomere? The sarcomere is just these repeated subunits of actin and myosin filaments, and it's really important that you understand that a sarcomere is the unit of contraction, but there are many sarcomeres in a single muscle cell. Okay, so that's everything that you need to know about the structure of the sarcomere and the structure of muscle. Well done.